Welcome back to BiochemSerye Learning on the Go. I teach biochemistry at the college level, and today we're doing a deep dive into podcast 2C.
Chemistry of disaccharides.
That's right, chemistry of disaccharides.
And I'm the other host. You can usually find me teaching biochemistry to med students.
We're going to unpack some key concepts about disaccharides.
Those fascinating molecules that bridge the gap between simple sugars and complex carbohydrates.
You know, when I'm teaching about sugars,
Yeah.
I always find disaccharides to be a real sweet spot. Pun intended.
Right.
They're complex enough to be interesting, but not so overwhelming that students get lost in the details.
I agree. They're the perfect example of how small changes in structure can lead to big differences in function.
Exactly.
And that's something we'll be exploring today as we break down different types of disaccharides and see how their unique features play out in biological systems.
Awesome.
So what do you say we start with the basics? What exactly makes a disaccharide a disaccharide?
Well, as the name suggests.
Yeah.
It's all about those two monosaccharide units linked together.
Okay.
It's like taking two simple sugar building blocks and snapping them together to create a slightly more complex structure.
Yeah.
They're still crystalline, water-soluble, and of course, sweet to taste, just like their monosaccharide counterparts.
Exactly. But it's that linkage between the two monosaccharides, the glycosidic bond,
Right.
that really makes disaccharides special. The type of glycosidic bond, along with the specific monosaccharides involved, dictates the disaccharide's unique properties.
It's like having two Lego blocks. You can connect them in different ways to create different structures.
Yeah.
And just like with Legos, those different connections in disaccharides lead to diverse functions in biological systems.
Perfect analogy.
Right.
Now, one way to categorize disaccharides is based on their reducing ability,
Okay.
which hinges on the presence or absence of a free anomeric carbon.
Okay, let's break that down a bit.
Sure.
When we talk about an anomeric carbon, we're referring to that special carbon in a sugar molecule that's part of the carbonyl group.
Yeah.
It's the carbon that can switch between a linear and a cyclic form.
Exactly. And if that anomeric carbon is free, meaning it's not involved in a glycosidic bond, it can participate in redox reactions. This ability to reduce other molecules is what defines a reducing sugar.
So we've got reducing disaccharides,
Right.
which have that free anomeric carbon ready to react.
Yes.
And then we've got non-reducing disaccharides where both anomeric carbons are tied up in that glycosidic bond.
Precisely. And understanding this distinction is crucial because it affects how these disaccharides interact with other molecules, how they're metabolized, and even their roles in certain biological processes.
It's like having a key that can unlock a door.
Okay.
Reducing disaccharides have that key, the free anomeric carbon, while non-reducing ones have their keys locked away in that glycosidic bond.
Now let's dive into some specific examples of reducing disaccharides. A good place to start is with maltose, which is made up of two glucose units linked by an alpha 1 to 4 glycosidic bond.
Hold on, alpha 1 to 4.
Yes.
It sounds like we're speaking in code. What does that tell us about how those glucose molecules are connected?
It's all about the orientation and the specific carbons involved in the bond.
Okay.
The alpha tells us about the orientation of the bond relative to the ring structure. And 1 for 4 indicates that the bond connects the first carbon of one glucose to the fourth carbon of the other.
So even though we're just dealing with glucose units, that specific linkage creates a unique disaccharide with its own set of properties.
Exactly.
But where do we actually encounter maltose in the real world?
Maltose is actually an intermediate product in the breakdown of starch and glycogen, those complex carbohydrates that serve as energy storage in plants and animals.
So when those long chains of glucose start getting chopped up during digestion, maltose is one of the products we see along the way.
Exactly. And speaking of digestion, there's a specific enzyme called maltase that's responsible for breaking down maltose into those individual glucose molecules that our bodies can use for energy.
Now, that's something our students can definitely relate to.
Yeah.
Those sugars we eat are ultimately converted into the fuel our cells need.
And to add a little fun fact to your next lecture, maltose is also found in malt, which is used to make beer and other malted beverages.
Now, that's a piece of information that's sure to get their attention.
Aha!
Okay, so we've covered maltose.
Yeah.
What's next on our list of reducing disaccharides?
Let's talk about lactose, the sugar found in milk. It's composed of one galactose and one glucose unit, joined by a beta 1 to 4 glycosidic bond.
Wait, beta 1,4.
Yeah.
So even though it's still a 1,4 linkage like maltose, the beta means there's a different orientation involved.
You got it. That subtle difference in spatial arrangement has a big impact on how lactose is digested. To break down that beta-1,4 bond, we need a specific enzyme called lactase.
And that's where lactose intolerance comes in, right?
Right.
If someone doesn't produce enough lactase,
Yeah.
they can have trouble digesting lactose, which can lead to those uncomfortable digestive symptoms.
Precisely. It's a perfect example of how even a small change in disaccharide structure can have real-world implications for human health.
Okay, we've got one more reducing disaccharide to cover, isomaltose. What can you tell us about this one?
Isomaltose, like maltose, is made up of two glucose units, but this time they're linked by an alpha 1 to 6 glycosidic bond.
Okay.
It's also an intermediate product of starch and glycogen digestion, but that different linkage means it requires a different enzyme called isomaltase to break it down.
So even though we're still dealing with those familiar glucose building blocks, that 1 to 6 linkage creates a unique disaccharide that our bodies handle differently. It's fascinating how those subtle structural variations can lead to such diverse metabolic pathways.
Absolutely. And it highlights the importance of paying attention to those seemingly small details when we're studying biochemistry. Now that covers the reducing disaccharides. Are you ready to move on to the non-reducing ones?
Let's do it. I'm especially curious about sucrose, the most famous non-reducing disaccharide of them all.
Ah, sucrose, the star of our sugar bowls and the culprit behind those sweet cravings. Sucrose is composed of glucose and fructose linked by a glycosidic bond involving both anomeric carbons.
So it's like those anomeric carbons are holding hands so tightly that they're unavailable to interact with other molecules.
Yeah,
That's what makes sucrose non-reducing, right?
exactly. And that unique bonding arrangement gives sucrose some interesting properties. For example, the enzyme that breaks it down is called sucrase, also known as invertase.
Invertase. That sounds intriguing. What does it do?
Invertase splits sucrose into its component monosaccharides, glucose, and fructose, a mixture known as invert sugar. This is why honey, which is mostly invert sugar, is sweeter than table sugar.
Wait, so the way sucrose is broken down actually affects its sweetness? That's fascinating.
Yeah.
It seems like every detail about these disaccharides matters, from the specific monosaccharides involved to the type of glycosidic bond.
You're absolutely right. And that's what makes them so intriguing to study. They're like little puzzles, each with its own unique set of pieces that fit together to create a specific function.
So whether it's maltose from starch digestion, lactose from milk, or sucrose from that piece of chocolate cake, understanding these disaccharides is key to understanding how our bodies work.
Well said. We've covered a lot of ground today exploring the structures and properties of different disaccharides.
Yeah.
But before we wrap up this part of our deep dive, let's take a moment to recap what we've learned so far.
That's a great idea.
Yeah.
It's always helpful to revisit those key concepts.
Yeah.
So what would you say are the main takeaways from our discussion of disaccharides?
Well, I think the biggest takeaway is that disaccharides, despite seeming simple, are incredibly diverse.
Yeah.
We've seen how just two monosaccharides can create a range of molecules with distinct properties based on how they're linked together.
It's almost like learning a new language.
Uh-huh.
Once you understand the alphabet, in this case the monosaccharides and the types of glycosidic bonds, you can start deciphering the meaning of more complex words, which are disaccharides.
Exactly. And that's what makes teaching this topic so rewarding. You can see those aha moments when students start connecting the dots between structure and function.
Absolutely. But it's not just about memorizing structures. I always try to emphasize the real-world relevance of these molecules, like we discussed with lactose intolerance. Something as simple as a disaccharide can have a significant impact on human health.
Right. And let's not forget the evolutionary implications. The prevalence of lactase persistence in certain populations with a history of dairy farming shows how our ability to digest specific disaccharides has been shaped by our ancestors' diets.
It's amazing how biochemistry intertwines with history, culture, and even economics.
Now, shifting gears a bit, let's delve deeper into the energetic roles of disaccharides. We often focus on their breakdown into monosaccharides for energy, but their formation is equally important.
Good point. Building those disaccharides requires energy.
Yes.
Right? It's like an investment storing energy in a form that can be easily accessed later.
Precisely. Think about plants. They capture sunlight energy during photosynthesis and convert it into chemical energy in the form of glucose. But they don't just leave it as single glucose molecules floating around. They often link those glucoses together to form disaccharides, primarily sucrose.
So sucrose is like a little energy packet ready to be transported to other parts of the plant where it's needed.
Exactly. And the reason sucrose is so well suited for transport is that it's non-reducing. Remember, those reducing sugars are a bit more reactive, which could cause problems during transport.
So it's like packaging those precious glucose units in a secure container to ensure they arrive at their destination safely and intact.
And this brings us to another important point. Disaccharides aren't just about energy storage and transport. They're also crucial building blocks for larger carbohydrates.
Oh, right. We briefly mentioned starch and cellulose earlier. Those are polysaccharides, meaning they're made up of many sugar units linked together.
Exactly. And if you look closely at those structures, you'll see familiar disaccharide patterns. For example, starch. That energy storage molecule in plants is made up of repeating glucose units linked in a way that resembles maltose.
So it's like maltose is a recurring motif within that larger starch molecule.
Yeah.
It's fascinating how these simple disaccharides contribute to the complexity and diversity of larger carbohydrate structures.
And this highlights the importance of understanding disaccharide chemistry as a foundation for studying more complex carbohydrates. It's like learning the rules of grammar before writing a novel. You need to understand the basic building blocks to appreciate the complexity of the larger structure.
I like that analogy. It really
Yeah.
drives home the point that even though disaccharides might seem simple, they're essential for understanding the bigger picture of carbohydrate chemistry and biology.
And that bigger picture includes not just plants, but animals too. Think about the glycosylation of proteins, a process that's essential for a wide range of cellular functions. Many of those glycans, the sugar molecules attached to proteins, are built from disaccharide units.
So even at the cellular level, these disaccharides are playing crucial roles in how proteins fold, interact, and function. It's like they're tiny but mighty molecular players in the grand symphony of life.
Well said. We've covered a lot of ground today, from the basics of disaccharide structure to their diverse roles in energy transport and even cellular signaling. But the real test of understanding is application. So are you ready to put your disaccharide knowledge to the test with some review questions?
Bring it on. I'm feeling confident, but a little quizzing never hurts. Let's see what we've retained from our deep dive into the world of disaccharides. Hashtag TTS, the Deep Dive episode 2024-1230, part 3 of 3.
All right, are you ready for some review questions?
Absolutely. Hit me with your first question.
Okay, let's start with something fundamental. Can you explain the difference between a reducing and a non-reducing disaccharide
Okay.
and why this distinction is important?
I think I can handle that one.
Yeah.
It all comes down to that anomeric carbon.
Yeah.
That special carbon in a sugar molecule that's part of the carbonyl group.
Mm.
In a reducing disaccharide, at least one of the monosaccharide units has a free anomeric carbon, meaning it's not involved in the glycosidic bond
Okay.
and is available to participate in redox reactions.
Yes.
Non-reducing disaccharides, on the other hand, have both anomeric carbons tied up in that glycosidic bond,
Right.
making them unable to reduce other molecules.
Excellent. You've clearly grasped the structural basis for this important distinction. Now tell me, why does this difference in reducing ability matter in biological systems?
Well, we saw how the reducing ability of lactose,
Mm-hmm.
with its free anomeric carbon on the glucose unit, makes it susceptible to reactions like the Maillard reaction,
Yes.
which is responsible for browning in foods.
Right.
This reactivity can also make producing sugars more prone to oxidation, which can be detrimental in some cases.
Uh-huh.
Non-reducing sugars like sucrose are more stable and less reactive,
Right.
making them suitable for tasks like long-distance transport in plants.
Precisely. The reducing or non-reducing nature of a disaccharide influences its stability, reactivity, and ultimately its function in various biological processes.
Right.
You're 2 for 2. Ready for question 3?
Brain M.
Okay, shifting gears a bit. Can you describe the role of specific enzymes in the digestion of disaccharides?
Absolutely. We talked about how different disaccharides require specific enzymes to break them down into their component monosaccharides.
Yeah.
For example, maltose is broken down by maltase, lactose by lactase,
Uh-huh.
sucrose by sucrase, also known as invertase, and isomaltose by isomaltase. Without these enzymes, our bodies wouldn't be able to efficiently digest these disaccharides and extract energy from them.
That's a great summary. You've highlighted the specificity of these enzymes and their crucial role in carbohydrate digestion. Now, for question 4, how does the structure of a disaccharide influence its properties, and can you provide specific examples?
This is where it gets really interesting. We've seen how seemingly small changes in disaccharide structure can lead to big differences in their properties.
Yeah.
For instance, lactose, with its beta 1 to 4 linkage between galactose and glucose, requires the enzyme lactase for digestion, which explains why some people have trouble digesting milk. Sucrose, on the other hand, has that unique glycosidic bond involving both anomeric carbons, making it non-reducing and giving it properties that make it ideal for transport and storage in plants. And maltose, with its alpha 1 to 4 linkage, is a key intermediate in the breakdown of starch, which is a major energy source for many organisms.
Excellent. You've connected the dots between structure and function using specific examples to illustrate your points. You're clearly mastering this material. Now for question 5, can you explain how disaccharides contribute to the formation of larger carbohydrate polymers, and what are the implications of this?
We touched on this earlier with starch and cellulose.
Yeah.
Starch, the energy storage molecule in plants, is a polymer of glucose units linked in a way that resembles maltose.
Yeah.
So you could think of maltose as a recurring motif within that larger starch structure.
Yes.
Cellulose, the structural component of plant cell walls, is also a glucose polymer, but with a different type of linkage.
Right.
These examples highlight how disaccharides serve as building blocks for creating those diverse and essential polysaccharides we find throughout nature.
Precisely. You've recognized the fundamental role of disaccharides in building those complex carbohydrate structures. Now let's shift gears slightly for question 6. Can you describe a real-world application of disaccharide chemistry?
Well, the most obvious example is lactose intolerance, which we've already discussed. But beyond that, disaccharide chemistry plays a role in food science and technology.
Okay.
For instance, understanding the properties of sucrose and its breakdown products. Glucose and fructose is essential for developing sweeteners, controlling browning reactions in food processing, and even creating new food ingredients.
That's a great example. Disaccharide chemistry isn't just confined to textbooks and classrooms. It has practical applications that impact our daily lives. Okay, ready for question 7?
Absolutely. Fire away.
Let's test your recall. Can you name 3 reducing disaccharides and their constituent monosaccharides?
No problem. We have maltose, which is made up of 2 glucose units, lactose composed of galactose and glucose, and isomaltose, which is also made up of 2 glucose units, but with a different linkage than maltose.
Perfect. You've aced the disaccharide memory test. Now for question 8. Why is understanding disaccharide chemistry important in a medical context?
Well, besides lactose intolerance, which is a common medical issue, disaccharides play a role in various metabolic pathways and diseases. For example, understanding how sucrose is broken down and how its components glucose and fructose are metabolized is crucial for managing conditions like diabetes and obesity. Additionally, defects in disaccharide metabolism can lead to various genetic disorders.
Excellent. You've highlighted the relevance of disaccharide chemistry to human health and disease. Now for question 9. What are some challenges in teaching disaccharide chemistry to students, and how do you overcome them?
One challenge is that students often get bogged down in the details of structures and nomenclature. To overcome this, I try to emphasize the big picture and connect those structural details to real-world examples and applications. I also use visual aids, analogies, and even hands-on activities to make the material more engaging and relatable.
Those are great strategies. Making the material relevant and engaging is key to helping students grasp these sometimes abstract concepts.
Yeah.
All right, ready for the final challenge, question 10?
Let's do it. I'm feeling like a disaccharide expert at this point.
If you could leave our listeners with one key takeaway about disaccharides, what would it be?
I would say that disaccharides are a perfect example of how small changes in molecular structure can lead to significant differences in function.
Okay.
They highlight the elegance and complexity of biochemistry, and understanding them is crucial for grasping a wide range of biological processes from digestion and metabolism to plant physiology and human health.
That's a brilliant summary. You've captured the essence of why disaccharides are such fascinating and important molecules.
Well, I have to say, I've thoroughly enjoyed this disaccharide deep dive. Thanks for guiding me through this sweet journey and for sharing your insights.
The pleasure was all mine. You've been a fantastic student and a great co-host. And to our listeners, we hope you found this exploration of disaccharides as enlightening as we have. Until next time, keep those glycosidic bonds strong.